Alright guys, in our second lesson today we're going to learn about DNA replication and the cell cycle. We're going to take a look at the basics behind the process of DNA replication and how chemicals work to replicate DNA. There's going to be a little bit of a review as we're going to identify and point out some events of each stage and phases of the cell cycle, as well as to identify the 3 steps that's required for DNA replication. So let's take a look at DNA replication first. We learned this last year where for mitosis to happen, you have to have a copy of the DNA, and those chromosomes are the DNA. And that when that replicates, it gets itself ready for mitosis. So during that replication, 2 DNA molecules are made from 1. And the parent DNA molecule will have the exact same DNA as the daughter molecules. So they're going to be identical, and they have the same nucleotides arranged in the same order. So in DNA replication, since DNA always pair up, and we know that for the from last lesson where your nucleotides are going to be complementary. So we already know that if the strands were to split up, we know that free-floating nucleotides are going to go to a set location because they have to be complementary. So in DNA replication, one strand will act as a template to make a new strand. And in this situation, there's going to be 3 steps that goes into this entire process. The first stage is called unzipping and unwinding, and it involves something that is called a helicase. And helicase is an enzyme that breaks hydrogen bonds between the nitrogenous bases that holds the 2 strands together. So that is located right here. Okay, so helicase, generally speaking, anything that ends with ase usually is an enzyme. So if you look at chemicals in our body, there's like lipase, right? Carbohydrates. These things are enzymes that break down lipids. So lipase break down lipids, and carbohydrates break down carbohydrates. In the same way, helicase breaks down the helix. So when that happens, the double helix will unzip. And if you try to separate 2 strands of a string, it will most likely not. So this thing called gyrase is going to act as a detangler. So gyrase will also work at this time to make sure that detangling happens so that it doesn't get tangled up and the DNA can actually split properly. Stage 2, or step 2, we have complementation. So that's when your base pairs will come together. So in this area right here, you have 2 individual strands of DNA, and then there's 3 nucleotides that are going to join up with the complementary base pairs. When the nucleotides get attached, that's when they start to link. Okay, so then that's part 3 where we have linkage, where you have polymerase, which helps the free-floating nucleotides join up with the open strands of DNA. Polymerase comes from the word polymer. Polymer is something that's used to attach or to adhere certain things together. So this closes up both ends of the DNA, and this links the new backbones of the new DNA. So as you can see here, we have the polymerase, which attaches the nucleotides together, and as they come together, you have a new strand. And at the end, you're going to have 2 strands that came from 1. Okay, and that's the same concept as having a parent DNA and then 2 daughter DNA. So into our cell cycle, this should be nothing new to us because we learned this last year in grade 10. And in order to replicate, we know that eukaryotic cells have to go through an orderly sequence known as the cell cycle. And there are 2 main stages. One is a growing stage, we call that interphase, and the other where we have the actual cell division, that's called mitosis and cytokinesis. So a little bit more of a review on this. As you can see that the interphase is right here, takes part in this big chunk of the cell cycle, and it spends about 90% of the time in interphase because that's where it grows, prepares itself for the division of the nucleus and its organelles. And during interphase, the cell does all the regular cellular activities, and there they will grow in size, volume, and mass, and that's where they duplicate their DNA. Now, going into each part of the phases of interphase, we have G1, which is gap 1. They carry out all cell activities, all the metabolic activities that's required for the S phase. Okay, and then at the end of the G1 phase, cells either go into S phase or some sort of rest. During the rest phase, cells can still function, but replication does not happen. And when it gets into S phase, that's when it starts to replicate its DNA. We can see that the chromatin in the nucleus will begin to condense and become visible as chromosomes. And then it'll go into G2, which is gap 2, and it'll continue to grow and continue to function so that it can prepare itself into mitosis. And the last little bit where it's the M phase, which includes two processes. We have mitosis, which is division of the nucleus, and cytokinesis, which divides the cytoplasm. And if we look back at a few slides ago, we can see that this is actually the smallest part. The M phase is the smallest part of the cell cycle. It only takes up about 10% of the time that the life of the cell is going to be in. And now the timing of the cell cycle and the lengths of the different phases really depend on the type of cell and its environment. So when it is ready and when it has all the nutrients that it needs, then the phase will happen faster. And finally, we have cytokinesis, where it's strictly cytoplasmic division, and it's cytoplasm and all the contents besides the nucleus, because the nucleus is the mitosis process, but your cytokinesis is everything else. And it starts before mitosis ends telophase. And about half the cytoplasm that contains half of the organelles goes to each of the daughter cells. And as they split up, we can see that there's different process for animal cells and plant cells. For animal cells, there's a little pinch that happens right here in the middle, and it separates the cytoplasm into two equal parts, and as they separate, it becomes two cells. But for plant cells, there's a cell wall that gets created here, and that cell wall creates a division, and it turns into two new cells. Okay, so we just talked about DNA replication, the three steps of unzipping and unwinding, complementation, and linkage. We talked about the enzymes that are involved in the process and how the whole process is required to make two daughter DNAs from a parent. And we also reviewed a little bit of the cell cycle. In our next lesson, we're going to review some of our mitosis, and that'll lead us to meiosis next week. Okay, thanks for joining us, and have a good one, people.